And Karin, it's very good always to see you. Thank you much.
Likewise, thank you so much. It's always great to come back to be part of the Climate Center. And I'm really thrilled to be here today. I'm just trying to figure out, can you see the slides? Okay, perfect. So we are good to go. So I'm Karin Ardon-Dryer, and I'm very, very, very honored to be here today. I remember the last time I gave a talk in Science by the Glass, that one when we still were able to meet in person back in, I think it was 2019, when we actually sat in a bar and a pub and enjoy a glass of beer. But I'm still thrilled to be here today and to talk to you about why should we care about dust storms. So we heard earlier, yes, we have a lot of dust in here. That's the main reason I'm here. So let's really go down into how much dust do we have? What's the impact that it has on our life? And why should we care? So I want to start with a quick introduction just to put us all on the same page. When we talk about dust particle, we talk about aerosol particle. I'm not talking about those that have on a spray can, although they are considered as aerosols. Aerosol are basically solid or liquid droplets suspended in the air. Just to give you the sense of how small they are, I pulled up a single human hair of my hair, and you can see the curly that's here, just to see the size of it compared to a 1 cent. And if you look at the diameter of that piece of hair, now you get a real magnitude in terms of how small these aerosols are. So we will refer to them as particulate matter, PM, and we'll talk about PM 2.5, particulate matter up to 2.5 micron, or particulate matter up to 10 micron. Now these aerosols are everywhere. Even if you don't see them now in the room where you are, they are particles. When do we get to see them? When there's a lot of them in one time point in the air. So for example, if you look at the black smoke coming from the car in front of you, if you look on biomass burning, when you're cooking, there's some aerosol they emit, and mainly through dust storms. That's considered to be one of the main aerosol sources that we have, the natural sources that we have. Now it's important to know that these aerosols don't have boundaries. They move from location to location. They don't care about physical boundaries. For example, we have some dust particle here in brown moving from the Sahara all the way towards the US. And we're going to see some of them in a few more slides. So here's some definition that we need to know as we're moving into a dust storm. So first of all, dust event is a meteorological phenomenon common in arid and semi-arid environment when we have strong winds that allow for the particle to blow up into the air. Now you can see these two images, one we're taking here from Lubbock and another one in Israel, and just the reduction of visibility in those clean day compared to the storm or dust event day. Now, dust event, as I mentioned, we increase the aerosol concentration, we reduce the air quality and impact on human health. And we see that these dust occur everywhere. Here in the US, we also have them, not just here in Texas, but they are occur in many places around the world. The biggest sources of them is the Sahara in Africa. So why should we care about dust? Why should we care about dust in general? Dust has a big impact on many aspects of our life. Impact on agriculture, for example, it's known to destroy corn, little corn plants, and basically kill the entire year. It can impact on animals. There has been work showing that animals can get suffocated during dust event and die. There's an impact on human health. We're going to talk about it later on, but one example is valley fever, which is very common in Arizona and in California. These are fungi grown on the soil that can uplift during dust event, and once you inhale them, you get infected by this disease. Also, dust is very important for cloud formation, for precipitation. Without dust, we would not have clouds, we would not have precipitation. So they have a very important aspect. But again, going back to the negative impact, there's a lot of car accident. This happened just north of Lubbock back in 2019. This is also from Lubbock, from the airport. And this is in Utah when 22 people were piled and 8 people lost their lives in one dust event. Now, going back again to those terms, because it's very important that at the end of today, you will be able to catch the difference between dust storm and dust event. So a lot of people use dust storm, but truthfully, there is a difference between dust storm and dust event, and it's all based about the visibility. How far can you? A dust event defined by the World Meteorological Organization as an event that is visibility that reduces up to 10 kilometers of the distance. But a dust storm would be a reduction of visibility below to 1 kilometer, as you can see here in this photo, or as a comparison of this one. Another important thing that we need to keep in mind is the difference between dust storm and dust event. Dust event is something that you would have in a sea. If you walk in the sea and you felt particle hitting your knee, that would be sand particles. Sand particles are larger, so they cannot uplift into the air. But dust particle can definitely uplift into the air and basically move and travel over long distance. So here in the US, we like to distinguish between dust storms and haboobs. Truthfully, haboob is basically saying a dust storm in another language. It came to the US with American troops who were deployed in the Mediterranean back in the First World Gulf War. And they saw these type of dust there, and they brought it to the US. Here, we are really defining between dust and haboobs based on the meteorological causes. So we have synoptic dust events, which are mainly large scale, for example, a front, a cyclone, a drought, a low pressure system. These are larger area where we have a lot of dust that can cross the area, as you can see in this image that we had here back in 2019. A convective event is basically a storm. It could be a single cloud that we have 70, 100 kilometers away from us. And as precipitation comes down, they evaporate, they create wind, and they hit the ground and basically go to the side and uplift the dust particle or the sand that's in the surrounding, and we can see that wall of dust. So these are images now that I'm going to show you from Lubbock, from MCOM, where my office is, and you can see this wall of dust, the haboob, and this is another image of this haboob taken from Lubbock, and just the regular dust storm or dust event with synoptic continuous flow of dust particles. But it's interesting that while these are very distinct, different meteorological, sometimes we have seen dust events that had a combination of two, both of them. Sometimes one start and then the other one intensify. Or back in 2021, we had a synoptic dust event and a convective one. So we had these two causes happening at the same time. And these are just images of this convective haboob and how it looked from the ground. And this picture was taken by one of our Ph.D. students in our program. So you can just see the amount of dust that we had in the air. So as I came to Lubbock, I really wanted to know how many dust do we have here? And how many of them will be convective and how many of them will be synoptic? To try to understand if they have a similar impact on the environment, similar impact on air quality. So to do that, what we have done is we have taken one of our meteorological stations that we have here, the Aerosols station, of our automated surface observation system located at the airport. And this station collects measurement of temperature, dew point, relative humidity, pressure, wind speed, wind gust, wind direction, visibility, precipitation, a lot of the basic information that we would need. But also, because we have an observer at the station, which is very unique to our site, we also have the ability to get information on dust events. So these observers provide in the METAR, which is the report from the dust event, information if we had a dust event. In other places in the state, most of the sensors are automated, so they will not have indicates of dust, but they would have indicates of haze. And this is how this report would look like. So we have a lot of number indicating the different meteorological conditions and also the code representing blowing dust event. So we took data from our METAR from 2000 to 2019. In addition, after identifying each of dates that had potential to have dust event. We also looked at the area forecast report, which has information from our local National Weather Service, but they put information on each one of the dust events. Now, this is perfect for this area when we have very good people, observer and National Weather Service team that provide a very in-depth discussion. So for example, we look for keywords to tell us which type of dust it is, as a synoptic or maybe a convective. We can go on and look at radar to identify were there any storm nearby? Was it really convective? We can identify maps of pressure to see was there a trap? Was there a low pressure system? So we can look at all that information to identify dust event and their causes. So for that period of time, 2000 to 2019, we identified that Lubbock had 420 dust events. On average, that was about 21 dust events per year. Obviously, there were years with more dust events and years with less, but overall, we have a lot of dust. Most of our dust was synoptic. As you can see, 67% of our dust was caused by synoptic causes, and 30% was convective. But when we look on the distribution of these dust events as a function of month, this is very interesting. We see that we have most of our synoptic in the spring and most of our convective in early summer or summer. So these are very interesting because it allows us to distinguish between these events. Now, next we wanted to know, how long do they last? Do they last for several hours as we have in other places around the world? And actually, we found that most of the dust here, it doesn't matter if it was convective or synoptic, most of the dust lasts less than an hour. So not that long as we had. We did have some synoptic event that had 14-hour long reports, but overall, most of the dust lasts 1 hour or less. And we noticed that also the time of day, not just the season is different, but also the time of day that those dusts occur is different. Most of the synoptic occur in the middle of the day around noon, and most of the convective occur in late afternoon. So we found that this is very important information for us here in Lubbock as we want to now understand the impact on air quality. But a lot of people told me that Phoenix has way more dust than we do. And I said, no way, we have much more dust than they do. So what we did, and this is a project that's been done by an undergraduate student, he basically repeated the exact same work for Phoenix, Arizona. And in that case, we didn't use only one sensor, we used 10 Aerosols stations. So for Phoenix, only for 2005 to 2018, that was the period that we analyzed, we found that they had 469 dust events. Now, most of their dust in Phoenix are happening in the summer, and they last, again, less than an hour. When we analyze the cause of these dusts, we found that most of the dust events in Phoenix are convective and not synoptic. The frequency of synoptic was very low. So because they were a different period of time, we decided to compare. We took the exact period of time, 2005 to 2018, and compare between Phoenix and Lubbock. Unfortunately, we lost. Lubbock had less dust event compared to Phoenix. These are for the exact same period of time, but it's interesting to see the differences. These are the variation for you, which I have not shown you yet, that we found here in Lubbock, and these are the ones that we found for Phoenix. And this is the distribution per month. So our dust, most of it is around spring, March, April, and June. And for Phoenix, it's completely summer. That's very different. So with that information, we really want to understand what is the impact on air quality. The project related to Phoenix is still ongoing, but I can definitely tell you what we know about Lubbock. So just as an example, if you don't know, I'm originally from Israel, and in Israel, we have a lot of dust. Now, if you don't know where Israel here located, it's in the Mediterranean, and most of our dust come from Sahara, but it also comes from other locations. This is one example for one dust event, and this is how the air looked like, the air quality looked like in that day. And you can see here the concentration of PM2.5 in red and PM10 in blue as a function of time. The daily average were these values, And you can see that they are way above the World Health Organization or the EPA, which is the US Environmental Protection Agency values, both for PM 2.5 and PM 10. Meaning, if you were outside during this dust event, that was problematic. Keep in mind that when we are outside during a dust event or where there are higher concentration, whatever you're breathing can get into your lungs. So it's very important to know the concentration in the different time frames. So can we do the same thing for Lubbock or for Texas in general? So here in Texas, this is the spread of our sensor that we have. These are the only sensors that we have across Texas. And as you can see, there's a lot of gaps. Just to take West Texas as an example, Amarillo, Lubbock, and Odessa, an area of more than 100 square kilometers, has only 3 sensors, and all of them are only for PM 2.5. So we don't really have enough sensor, and we're hoping that this is something that will be able to increase over time. So let's focus on Lubbock. In Lubbock, we had one single PM 2.5 sensor, and we actually looked at the concentration of these sensor over the entire period that it's been active, which was 2011 till 2019, right before we published this in a paper. And you can find it here in the model. So the sensor was located in 3 times. So this is the first location, the second location, and the third location, which is now located right out on a loop at 12th Street. And this is the photo of that sensor, and this is how it looked like. So what we can see, the red values are just daily values, and the green are daily max. So overall, the red doesn't really pass this threshold of the EPA, which is 35 microgram per cubic meter. But you can see all those big green peaks when the maximum reach way above the threshold. So what does it mean? So let's see them. This is one of our biggest and famous dust events we had here in Lubbock. It happened in 2011, and I'm going to let you sort of absorb and see the amount of dust that we had. So these are the picture that we're taking here from MCOM on both sides of the building to show you the amount of aerosols and dust that we had. So we looked at the concentration of these sensors. So this is the concentration of aerosols that were happening. This is the PM 2.5, and this is a function of time. So obviously this increase happened during the dust event when we reached the station. But when we are looking at the daily values, this was the number, 23.5. Why I'm so emphasizing on this number? Because that number was below the threshold for World Health Organization and EPA. Meaning, if I'm only looking at daily values, I would think this day was fine. Nothing happened. But you saw the photos. People were really scared when that dust event occurred here in Lubbock. And we have similar dust like this every few years. And again, when you're outside and you're breathing those particles, they can get into your lungs because some of them are very small. So averaging across a day, massive fluctuation of the dust concentration. And in places like Texas or West United States, when we have a lot of dust events, we need to start looking at the impact in a different way. Here's another example. We're expanding to other parts of Texas. So this was the special event that was a mixture of the convective and the synoptic event. So you can see some of the dust here, and also here you can see it in pink, the synoptic and some of the convective haboob. So we looked at PM sensor across Texas from Odessa all the way to Houston. Now they had dust in that event, but also, as you can see, dust that came from Mexico. These are the increase in particulate matter during the time that the dust reached the station. And again, if I only average the concentration as a daily function, none of those sensors cross the threshold of the EPA. It might cross it with the standard deviation, but you would not even think that this day was problematic. So we have to find a better way to look at the impact or define the impact that dust events have on air quality. Also, as I mentioned earlier, there are not that many sensors in Texas. We only get for Lubbock information on PM 2.5. We don't have information on other sizes. We don't have information on the size distribution, which will impact basically on how many small particles we have during this dust event. So that was the main reason we decided to build AEROS. AEROS is Aerosol Research Observation Station. It's located here on campus on an electrical engineering building. And if you walk on the engineering path, mainly on Holden Hall building, you can look towards the electrical engineering building and see it. AEROS has been located in that location since March 2019, and it's been continuously monitoring the air quality in Lubbock since then. If you want to read more about it, it should come up very soon. Jeff was accepted to AMT, and it should come up online, the updated version, very soon. So you're very welcome to look at it. So what kind of information we get from AEROS? The information we get from AEROS is various. The main part of the sensor, which is the shed, has different instrumentation. And with those instrumentation, we can get information for every minute on PM, different sizes, PM 1, 2.5, 4, and 10, but also on the size distribution. And these are the concentration as a function of size, which basically tell us how many particles smaller than 1 micron we have, a size that can penetrate into the oxygen. So these are the different instruments. In addition, we have the Harvard Impactor setup, which you can see here. This is a setup provided by Harvard Impactor, where we collect particles on filters for PM 2.5 or PM 10. These are gravimetric measurements. They're done for 24 hours from midnight to midnight. And we can take the filter, we can analyze it in the lab, look at them under the microscope, send them for sequencing to look on the presence of some biological particle, things that I'm going to show you in a few more slides. So we had a chance during this period, ever since the station has been active, to measure different dust events. For example, we had one dust event that was synoptic dust event. And you can see a continuous flow of aerosols with the decrease of visibility and increase of wind speed. And this is the image for our blood of this dust event. We had a convective event, and you already saw that Haboub, with the sharp decrease in visibility, sharp increase of wind speed. And for those with some sharp eyes, you might be able to see this wall of dust, even in the radar. We had another convective event following that June 5th that happened in June 21st. And this one with, again, sharp decrease in visibility, sharp increase in wind speed. I'm going to show you the video in a second, but just keep in mind, the storm that caused it occurred about 70 to 100 kilometers away from us. And how this dust looked like? Interesting. Sorry. My computer is excited to show you something. So here it is. All right. So I have this yellow line. I guess the computer decided to be excited about this talk and added that. But this dust event also occurred. And basically, we had the ability to measure it and to look at it over a period of time. So when we look on, and I'm sorry about this yellow line that just happened on the screen. Let me see if I can stop share. All right, and I'll share again just to be sure you can see it correctly. There we go. So when we look on the daily value of these dust events, the synoptic and the two convective, we can see that the daily value doesn't really pass the threshold. But look at the standard deviation, how big it is. And that's part of the problem. We never look at the standard deviation. We always report just the daily value. If it's above the threshold, below the threshold. So we should start thinking and putting emphasis on these information. Another thing that we wanted to examine is how the aerosol concentration change as a function of time. So we have here different PM sizes, and this is the synoptic event. It lasted for 12 hours, and these are the two convective events that lasted for only 20 minutes or 30 minutes. Now, if you look at these values as a function of 1 hour average, it seems as if the convective events were not that important, didn't had a lot of concentration. But it's actually the way you look at the data. Because they lasted less than 20 minutes, less than an hour, when you're averaging across an hour, you're misrepresenting the actual impact. Because when we calculated this event as if every 10 minutes, as a function of average of 10 minutes, now you can see the true impact of this convective event that actually had a much higher concentration even than the synoptic event. But we need more measurements. We need to identify if this is really something that's happening in all our convective events, or maybe only in those that actually form hubbles. So there's a lot of work for us to do here still. We also looked at the impact of psi distribution. Now, why is this important? Because when you talk to people who work on dust, they would say, oh, dust, these are large particles, you shouldn't worry. But actually, we should, because we see here the concentration of particle as a function of size. These are the measurements right before the dust occurred. The light gray are daily average, and the black one are at the dust peak. So you can see that all of those dust events, the synoptic and the two convective, basically had an increase of particles, even particles smaller than 1 micron. So they have the potential to penetrate into our lungs if we are outside during these events. And these are just a comparison of these three different particles or three different dust events, because we're really interested in understanding how similar they are and how different they are, because we want to be able to distinguish their true impact. So we took the particles that we collected during this dust event and we examined it under the microscope. And we found that most of the particles contain elemental compositional similar to dust particles that we're familiar with. But also, we found some nice biological particles, some pollen on our dust particle sample. So that actually started an interest of how many biological material do we get during these dust events. So a week after this dust event, we collected another filter exactly the same way for the exact same period, and we sent both of them for sequencing. We also sent a clean filter. And we found that there's a very high concentration of bacteria of different types and fungi that basically increase during these dust events. And as you can see, they're much higher than the clean day or even the clean filter that we sent for sequencing. So these are the 10 most abundant bacteria and fungi. And if some are you familiar with some of those names, there are known studies that some of these fungi are very problematic for our health. But here in Lubbock, we don't only have dust that's coming from our area of West Texas, New Mexico. A lot of time we have dust that actually travel across the world to get to Lubbock. And this is the example we had back in June 2020. If you remember, there was a big dust event that everyone referred to Godzilla. It was a big dust event that traveled from the Sahara all the way to the US. And we can see here a back trajectory now for Lubbock, showing that the air path actually came from Africa. So we took all the sensor in Texas, and we examined each region to identify what was the impact of this dust traveling from Africa on people who are living here in Texas. And we separate these impacts by different locations. And we can see we have here Houston in the center, Austin, Dallas, San Antonio, south part of Texas, Odessa, Amarillo, Lubbock, and also El Paso. Now, El Paso was not impacted by this dust event. They had some of their local dust. But basically what we found is that the air was coming in and doing a turn right in Lubbock and going back to the east side. So we see that there's an increase in concentration of particulate matter in each one of those sites, exactly on the time we found that these particles reached the location. So let's focus slightly about Lubbock. Here in Lubbock, at the time, that was during COVID. So the station was basically shut down. So we took the station, I took the station home, and I basically continued to measure the particle at my house. These are just pictures of the backyard, and you can see, and we also collected some particle and filters. So we found that even here in Lubbock, there was an increase in concentration during these dust events, and you can see some of these events here. We noticed that the concentration during the Sahara event increased the concentration of small particles. And also when we examined some of the mineralogy, we found some differences, but truthfully, because we have so much dust, it's hard to say how much of it is local and how much of it is actually coming from Africa. So we're still working on that aspect. So I hope that I convince you that these particles that we have here in the air could be problematic. And let's talk about some of their health implications, because these are something that we're doing here in the group as well. So small particles have the ability to penetrate deep into your lungs. We have some natural mechanism that's supposed to help us not even get those particles in. The hair in our nose, the eyelashes, mucus. If you have the sense of throwing up or spitting out during dust event, these are cleaning mechanisms that you might have in your throat. Now, large particle can penetrate into the upper respiratory, to the nose. Smaller will go into the upper respiratory area, and some of the much smaller one can go deep into your lungs. Now, here are some examples of lung tissue that were taken from American soldiers who were deployed in the Mediterranean. These are years after they came back. And you can see the accumulation of particles in their lungs. So once these particles are in, it's hard to get rid of them. And we can talk later about some of our natural mechanisms. So how do we study the impact of dust on health? We can do epidemiological studies, which are basically studies that based on statistics. How many people came to the ER? How many people had COPD? How many people took asthma medication? So mainly statistical. We can do in vivo, which means exposing animals to different dust particles and look at the impact, or in vitro, taking the dust, exposing it to cells and look at the health implications. Now, when it comes to the epidemiological studies, most of the studies show that there are some health problems, different types of health problems. But unfortunately, it is still not clear, is all the dust toxic in the same way? Because some of the literature showing that dust is not as toxic as we thought. And this is a very interesting question. We really want to find what makes one type of dust toxic and other don't. So this is one of the things we're doing in the group. And we're doing it in two different ways using in vitro method. One is the old-fashioned way, which is cell population. We're exposing the cells on a plate to multiple dust concentrations of a known dust type. And at a certain time point, 6 hour, 12 hour, 24 hour, we're looking at how many cells die. The other technique is a single cell method. The single cell method is basically a method that I developed at Harvard as a postdoc, and I brought it to Texas, where we're looking at every cell individually. Like, we're different, so are cells. And understanding the behavior of each individual cell will help us to shed light into this impact. So what are we doing? We're using A549 cell. For those who have not taken a cell biology class for some time, this is the cell nuclei where we have most of the genetic information, and this is the cell cytoplasm. We're going to see an image now. This is what we look at. We see the cells moving. We see the interaction of cells. We can identify when the cells divide. So using this technique, we can look and monitor the behavior of each individual cell over time. We can identify the interaction of each cell with the dust particle. We can detect the exact time of death. We're taking images every 15 minutes. So we know exactly when the cell died during that period of time. And we can also identify the type of death, which you'll see in a second could be very meaningful to understand the impact on health. So if you want to hear or read more about it, here's some information on the paper that basically describe this method. And there are more paper now under the pipeline, under preparation, to show you some of the other findings that we have. So we normally take different wide concentration of aerosol particles. Because we want to understand when would it be toxic, and would it be the exact same concentration for different type of dust particles. So when we look at low concentration of dust, we can identify the cells and identify the dust particle in white. When we increase the concentration above a certain threshold, we cannot see anything. And that's the main reason we added a nuclei marker, a fluorescent protein, into our cell. And now when we're monitoring and adding dust particles, we can still identify the cell and look at their behavior. And by looking at the nuclei, we can identify if the cells remain alive or if it's dead. And let me show you that. So cells that remain alive basically maintain the shape of the nuclei. Cell can divide, but overall, the cell will stay at its shape. So this is a cell that remains alive. But sometimes we'll see this. These explosions are basically type of death known as necrosis. So we see that the cell changed the nuclear shape, not to a division mechanism, but it's basically exploding. There we go. And this is a cell death. We also notice that the cells are basically doing something very interesting. So here's the cell, here's the nuclear marker, and you can see the particle here, and the cell is basically engulfing those particles. And you can see that at some point we started to see a change in the nuclei shape. The cell is going to grab some more dust. It's changing the nuclei shape, and then it's going to die. So it's dead. So we notice that when when the cell takes more dust particle, it increased its chance to die. And it's very important to understand that factor, because we still don't know why these cells supposed to taking those dust particles. They're not supposed to do that. But also understand what impact that may have. So here's again another view to look at the impact. If you can look here at the center, you can see the dust and how the cell are basically engulfing those particles. So as I mentioned, when we expose the cell can either remain alive or die. And when they're dying, they're focusing on two types of death. Apoptotic, basically the cell maintains all the information inside of it. Nothing bad will happen to us health-wise. Or necrosis. Necrosis is those explosions that I showed you earlier, meaning that the cell will release everything into the bloodstream. Now here's some images showing you how it looks like. You see the explosion, and we saw some of apoptotic bodies. So overall, we found that as we increase the concentration, the percentage of apoptosis decreased, but the percentage of necrosis increased. Now this is very important information, because we want to know what is the threshold that makes some of the dust particles to become toxic. Now, just as an example, we have been measuring some of the dust that we have here in Lubbock. Now, very recently, we added a new component into our station. This is the Anderson instrument that basically measures filter in the air, but it measures all the sizes. We call it TSP, total suspended particle. So during the month of December and January, recently, several months ago, we were able to measure during a dust event, and you can see the sort of brownish color of the filter, and during a non-dust event, relatively clean, and you can still see we have still some particle just because of all the transportation and the fact that we live in a city. So for this experiment, we used the cell population experiment. The cell population, as I mentioned, is one of the well-known methods. We basically use the exact same cell. We plate the cell on a plate at a known concentration. We add the particle into the plate. This is how it would look like before we added the particle or after we added the particle. And 24 hours after the exposure, we extract the particles, we take the cells, we count the cell to basically see how many cells remain alive and how many die. So just as a comparison, these two events, we found very interesting findings. These are the concentration or percentage of cell death as a function of concentration. And this is the dust event and the non-dust event. A very interesting aspect is that we had a lot of fungi in both of the events. The filter was full with fungi. And I don't know if you can see some of the fungi here. When we had some attempt to run a single cell experiment, we were just unable to do that. Another interesting factor is that for the dust event, only the 500 microgram per milliliter was significantly compared to the control. We don't have that comparison for the non-dusty days, so we don't know if it was higher, if they would have been different. So we are hoping to continue to collect some of the data to understand better what is happening here in Lubbock. And again, look at some of the fungi that we have here, some of the bacteria, maybe some viruses, but also the chemistry of the particle. So What we're doing in addition, and this is a project that actually finished, but we're now trying to wrap up into a paper. We have a large samples of dust from all over the world. And these are just 17 soil samples that were collected in Africa. And you can see on the map where they're from. And just the picture at the bottom shows you that they have different colors. So they have different minerals. So what we have been doing is we've been looking at the impact of these particles at different concentration on cell. And very interesting enough, if we compare some of these samples, we see that one sample located from Morocco had a relatively low impact on the toxicity. Another sample had much higher impact on the toxicity, where many more cells died. A third sample in Chad had a different behavior. Most of the cell death happened in 100 micrograms per cubic meter. Now, this is interesting. This is very similar to one of the work we've been doing in the past. So if we took all the sample and compared them at the 5 microgram per cubic milliliter, we see that there's some variability. Not all the dust is toxic. Some dust are not so toxic, some dust are more toxic. And we really want to try to understand why. So just as an example, we've been looking at 4 dust events. And the first thing we did, we wanted to look on the impact of sizes. So we compared the size distribution of these samples, and we can see that the most toxic one actually had lower concentration of small particles, which was very weird for us. Second, we analyzed these sample under XRD, or single particle in microscopy in SEM-EDX. And we then compare the composition or concentration of different elements to the percentage of cell death. So this is one example when we have the percentage of elemental composition as a function of cell death. We can't really find any strong correlation. We thought we would find that some element would be more toxic compared to others, but we can't. Even when we look at the mineralogy itself, comparing different quartz, carolinite, elite, or even just clay, the percentage of clay in each sample, we still don't find very strong correlation, meaning there's still a lot of work for us to do. There's still a lot of work to try to understand what makes some of these dusts more toxic than others. And this is still a project ongoing. Now, if you are, after this talk, still interested in hearing about dust, being part of a dust community, I want to be sure that you will get familiar with DANA. DANA is Dust Alliance for North America. This is a new alliance that was launched earlier this year. You're very welcome to register, scan the barcode, or check our email at dustalliance.na@gmail.com. Feel free to send us an email. We have a webinar once a month on the second Friday. And this is a very great community of people who are interested in the dust in general across the US, but also in Canada and some parts of South America. So if you're interested, feel free to join us and be part of DANA. And I would like to basically acknowledge all the students who are working so hard on these projects. And most of the work you have seen today would not be possible without their help. So some of the master and graduate and PhD student and the undergraduate student who work in the group, but also some of the work you have seen today were a former master student who graduated. Also, I want to take the opportunity to thank the CH Foundation, and thanks to them, we are able to do a lot of the cell microscopy project, and to Professor Kender from Darmstadt, who provided us a lot of the soil sample from Africa. And thank you for your attention, and I'll be happy to get questions.